Good afternoon, students. Good evening, rather. We are here for the second day's lecture in this Monsoon lecture series. And today we have a single lecture from Dr. Shomik Agarwal. He is the Assistant Professor and Head, Department of Zoology, Gaur Mahavidyalaya, Malda. Okay, and he is going to talk about the basics of immunology. It is going to be a very, very interesting class. I welcome you all, and I welcome HOD, Department of Zoology, Gurubadhan Government College, Dr. Ritesh Bishwa. He is going to start the proceedings for today. Then I'll be following up.
Thank you, Sandeepan sir, and once again, welcome sir. Welcome Swami Agarwal sir. It's our pleasure to have you with our college once again. My dear children, please make use of today's session. We have the honor of having Dr. Swami again with us. Last time also he joined for this special lecture we had arranged before. And on behalf of Department of Zoology, Gurubadhan Government College, I welcome you, sir, to our Monsoon lecture session. We have been trying to organize a little bit of this type of activity so that our students can be enriched with the knowledge of expertise like you people. And I hope today's session will also be going to be very useful to them. Thank you, sir. Please.
Thank you, Dr. Bishwa. Thank you, Shundipan sir, for inviting me. The entire, whatever it is it's an honor to me that you invite me again and again, and you all the time you speak so highly of me. I don't know whether I'm worthy of it or not, but anyways, thank you for your kind words. So, dears, yeah, please, sir.
Yeah. Sorry to interrupt you.
No.
I request any one of our students to change the screen. It is my great honor to introduce to my students. You already know him. You have done his class already. But again, it is my pleasure to introduce you formally. Dr. Shomik Agarwal. He is presently the head of the Department of Zoology, Gaur Mahavidyalaya, Malda, and he had done his master's and PhD from Shiksha Bhavan, Vishwabharati University, and he is an excellent researcher. He had been awarded with many fellowships and grants, only to mention a few. DST Fast Track Young Scientist, DS Kothari Postdoctoral Fellowship, Senior Research Fellowship CSIR, University UGC Fellowship, and possibly the most prestigious postdoctoral fellowship at Georgetown University, USA. Today he is going to discuss about the very intricate topic of human physiology, its introduction to immunology. And he has been one of my very close colleagues. We have collaborated in many projects, both academic and administrative, and I have learned a lot from him. And today I am looking forward to learn a lot of things also, and I also wish that all my students are going to enjoy his fantastic talk. So from here, I would respectfully request Dr. Shomik Agarwal to start presenting his lecture. Respected sir, please.
Thank you very much, Dr. Talukdar, for your kind words and everything. So, with your due permission from you and Dr. Bishwa, I would like to start then my talk then, if you have permission.
Priti, you please stop sharing so that sir can share.
Okay, thank you. So, without any further ado, I will just jump into the topic, okay? Because I have just 45 minutes. I will give you today what I am going to discuss is the basic of immunology. I will not go into the detail of it because you all know that immunology is a huge subject. And with any physiological abnormalities or activity, you can somehow link the uh, the immunology subject, the topic. So it is such a vast topic, subject, cannot be dealt in a year as well, I think. It is so huge. So, being all of you, being an undergrad student, what when I was discussing with Dr. Talukdar, I felt we can cover up the immunology portion, the basic of immunology, so that whenever you will read any book, it will be easier for you to understand. So, here I will try to give you a glimpse of what is immunology, the basic of it, so that you don't face any problem. Basic can be, I will just help you to understand the basics of immunology. Okay? If you have any question, any point of time, feel free to stop me. It is not for the first time. I think we have met before. I think. Is it so? Dear students, have we
Yes,
met
sir.
before? Okay.
Yes, sir.
So you can stop me anytime you want. And if you have any queries, please let me know. Okay? Don't feel hesitant.
Yes, sir.
Okay, so I'm start presenting my slide. Please let me know if my slides are visible.
Yes, sir.
Okay, thank you. Can you see a photograph of a fort?
Yes, sir.
Yes, sir.
So this is immunology. How come? Here, the fort, it's us, you, me, and whatever, the animal we are discussing right now. So, for the sake of simplicity, just consider it's us, you or me, anyone. This is our body. And all around, there are hundreds and so whatever. Anyone can come and attack. And so the fort has its own defense system. This story of attacking and how to prevent that attack, that is that will be the discussion of today, and that is immunology. Just think, or suppose, as I said, this is our body surrounded by water here. I am just telling you a story. Whether you can relate that or not, you just tell me, okay? Suppose some dacoits or the enemies are coming and they will attack. Where they will attack first? Here. Or suppose not suppose, it's an door. It will try to enter through this door. So, what will be the defense mechanism? It will have sentries here. It can come and they will try to break this wall if this is not open. If this door is not open, what they will try? They will try to break the wall and they will try to enter the fort, an enemy. So, how to defend that? They will have a stone or very thick wall so that cannot easily be broken. And if someone comes nearby, what will happen? These are watchtowers. This round shape are watchtowers. So the sentries are always there. They are moving all around this wall. We have seen in movies, right? The sentries walking all over this wall. The sentries watching from here. Guards are watching from here, some holding spears, some holding bow and arrow. Even somewhere you have seen it in some movies, they have bucket full of hot oils. If they come nearby, they can pour it. Right? Yes or no? Have you seen that in movies?
Yes, sir.
Yes, sir.
So, yeah. So I have mentioned three things. Just try to remember this story. It will be easier for you to understand the basic of immunology. Just hold on. ম্যাডাম আমি একটু ক্লাসে আছি, আমি পরে আপনাকে কল করছি। ঠিক আছে। Extremely sorry for the disturbance. So I have said the enemies can enter through the door, enemies can try to break the wall, enemies may try to climb the wall, and how to prevent it? By closing the door, by making her very strong wall, thick wall. Second, third, by putting sentries all over the place. Third, and they may hold some spears, bow and arrows, or hot bucket full of hot oil. Right? These are the things I have mentioned. And the first line of defense is this water here. Water is what you call that, all if that water is called moat, right? If I am not wrong, around the fort. These are the defense mechanism you can watch over here. So I will discuss one by one according to this picture. Suppose two types of immunity we have heard or read from class 8 or 10, right? That is innate and acquired immunity. All of you are familiar with this term?
Yes, sir.
Innate? Okay.
Yes.
So what is innate immunity? Sorry. Innate immunity is that immunity which is you are having naturally. That is innate or inborn. And acquired, which you have acquired through experience or through time. These are the two things. Very simple. I have said that our body, I have compared our body with this fort. And what I have said, the enemies are all around. Just imagine yourself. You are breathing. Dust particles, viruses, what not, that's entering your not entering through your nostril. Anything it can enter. If these things can enter through nostrils, just imagine the surface that is exposed, that is our skin. You must have heard someone that got burned and having serious implications from the burn because of that burn. Why? The skin is damaged. You know, most of the cases, a burn patient die not because of that burn, because of other infections. Are you aware of that?
Yes, sir.
Why so? Could you please tell me why they die of other infections, not because of the burn itself?
So the defense mechanism is like destroyed, so that's why.
So is it the only defense mechanism? How the defense mechanism is destroyed? Skin is only one. There are so many things, antibodies, B cells, T cells, macrophages, so many things are there. So why, if the skin is burned, that it will have its serious implications? Why? Why do you think it is having serious implication? What is your thought? I just want to know, just like a story. If it is wrong, no issues. Anyone want to give a try? Uh, okay, let me tell you then. See, 90 to 95% of infection, whatever or the germs all around, the skin is the first line of defense, and it stops 90 to 95% of all the germs all around you. Do you understand? Do you understand? It stops more than 90% of the infections. Then, if that system, that skin, is not there or it is defective, that can definitely have serious implications. Just imagine whatever the cells we talk, that acquired immunity we talk about, they just deal with 5 to 2% of infections. Most of these are inhibited in the first place. So very little is dealt by the acquired immunity. Am I clear to you?
Yes, sir.
Okay. So what are the defense mechanism? As I've already mentioned, the skin. And what is, why skin is a defense mechanism? Why just normally the skin is not broken, anything? Why can it cannot penetrate? Because of its structure. It cannot go inside. It has low pH. Sebum glands are there. They produce sebum. Their pH is controlled. Bacteria or pathogens cannot enter. Generally, it cannot enter. Okay. So that after that, we have so many openings. Nostrils, that is the nasal path, the gastrointestinal tract, the urogenital tract, eyes. These are the places through which the pathogens can enter. But we have here the mucus layer. Just imagine some oil, thing, or the water all around the fort. If, or imagine the wall is layered with some oil. So what will happen if the pathogen or the enemy tries to climb up the wall, this wall, it won't be able to climb up because there is a layer of oil. Similarly, here we have mucus layer. Mucus layer doesn't allow the pathogen to get hold of the system, and it helps to remove that from our body. Do you understand?
Yes, sir.
Okay. Then, what else do we have? Some mechanical action. Sneezing, coughing, peristaltic movement. Peristaltic movement is just where you have heard about these peristaltic movements in our gastrointestinal tract, right? One of the you always discuss. From the, just to give you an example, do you know, basically sometimes we have, you must have heard that a pregnant lady feeling, having the feeling of nausea, they sometimes they vomit, right? You must have heard or you have seen in movies. They have the pregnant lady having the sensation of nausea. Why so? Do you have any idea why a pregnant lady they're having the sensation of nausea or they are having that problem of vomiting? Do you have any idea why?
No, sir.
Okay. A theory kind of thing is that a protection of the baby. If the lady having too much of intake or having something toxic, it will create problem for the baby that is developing in her womb. So sometimes it's a theory kind of thing. Sometimes it is believed that system, the body has created that nausea kind of feeling, so it will vomit, no extra food will go, or if there's anything toxic that will be out. Now it's a debate. Some says it will create another problem. Definitely, if one cannot eat, having the feeling of nausea all the time, So that is another issue. But the system wants to give protection. It is also kind of protection for the babies. So peristaltic movement. So when you vomit, so there is a retroperistaltic movement. Peristaltic movement that goes, the food goes inside. If it is retroperistaltic, then the food will come out, right? So it is a mechanical kind of, when you sneeze with a full force, the air comes out with a gust, and if something is there inside your mouth or inside your nose, that will come out. Sneezing, coughing. When you cough, it comes out of your lungs or that portion. So these are mechanical and a line of innate immunity, a portion of innate immunity. So what I have discussed, skin, then it comes the another thing, that is pH, then comes temperature. All these are part of innate immunity. If can you tell me a portion where the pH is very low and it uh, helps us in protecting, or helps to protect us from the pathogen, where you have seen low pH that kills
Stomach.
very good, very good. Where else? In the urogenital system, right? In female urogenital system, it is pH is low, right?
Yes, sir.
Okay, chalo. So the, as you just mentioned, stomach is the excellent example where the pH is very low. Most of the bacteria or the pathogens get killed, and there are several other examples. So these are all part of innate immunity. What will happen if this, now so far these are all the mechanisms I have described. Now the wall is there, the slippery wall is there, the water is there that is helping to protect the fort. Then the doors are closed, they have something, many things. Now the intelligent pathogens are also there. They have special mechanism to climb over this wall. They have special tools to drill through this wall. They have, some can disguise among these people and can enter into the fort. Yes. So what will be the response? What will happen then? Anyone? What will happen then? If something enters the fort, suppose it's a real fort, what will happen?
Then the other person will try to fight.
What the other person? The sentries, the guards, right?
Yes, sir.
Yes. Here also the same thing happens. Where? In our system. There are few cells like macrophages. There are many other cells. I am not going into the discussion. Okay, natural killer cells, those are many cells. As I have already mentioned, today I will give you an idea that will help you to study other books because of the limited time. Now, just imagine 3 cells I'm talking about. B cells, I'm talking about dendritic cells, I'm talking about macrophages. Why? What are the specialties about these 3 cells? These are also called antigen presenting cell. What? Repeat after me. Antigen presenting cells, or in short APCs. You will find throughout your textbook APCs. Antigen presenting cells. Okay? So there are only 3 antigen presenting cells. What are those? What are those? B cell, dendritic cells, and macrophages. Do you get my point?
Yes, sir.
Yes, sir.
Okay.
Yes, sir.
Yes, sir.
So where these cells reside? Where these cells reside? Just imagine. Can you see my hand? So suppose this is the outer surface of the fort. A pathogen manages to enter through the wall inside. So we must have sentries here, and those sentries, out of many, I'm just mentioning a few for the sake of simplicity. Suppose a macrophage, for the simplicity. The macrophage dendritic cells all are here. They are roaming around. As soon as they get glimpse or they can see the pathogen inside, it comes running towards it and catches it. Okay? Arrest it. And what they will do? They will take no risk. Simply they will eat it up. What they will do? They will eat it up. Do you get my point? Anyone? All of you?
Yes.
Those cells will eat these pathogens, and so how they will eat it? They will phagocytose it, and inside they have some enzymes. Through these enzymes, they will simply chop it. Suppose they have the pathogen inside the cell, they will chop it. So, making hundreds of pieces out of it. Now, what will be its role then? Suppose, imagine, in true sense, you have arrested an invader. You, being a sentry, you have arrested it. So, first, what will be your duty? To kill that enemy, or to tie it up so that it cannot make any harm. First. Second, your duty will be to alert others. See, enemies have tried to enter the fort or have entered the fort. I have managed to catch one or two. So be prepared and get yourself ready so that you can find the others, find other enemies, or if it enters, then you can catch it. Yes or no?
Yes, sir.
Do you get my point?
Yes, sir.
Okay. The similar thing will happen here also. As I have mentioned, only one cell, the macrophage, what it has done? It has captured the pathogen. After capturing the pathogen, it will chop it or cleave it into very small pieces. Then its role will be to alert others. And what they will do? It is like that they are very rough. Imagine, you must have seen Kalima, the photograph of Kalima. What in that picture you will find, Kalima is holding the head of a a criminal, right? Similarly, here the cell will display those small or chopped pieces. On their surface through, just note it down, through MHC2. Right now you don't have to understand MHC. Through MHC2. Suppose MHC is hand. So it is exposing, it holding the chopped piece of those and the pathogen in their surface by holding this hand. We are calling it MHC2. MHC2 is available only with APCs, antigen presenting cells, are available or present in antigen presenting cells. So if there is a 0.5 number of question, where do you find APCs, MHC class 2 molecule? You have to answer in antigen presenting cells. Am I clear?
Yes, sir.
And all other cells in all other nucleated cells, they have MHC1, major histocompatibility complex 1. So where do you find MHC1? In all nucleated cells. But in APCs, you find MHC class 2. It's a very simple thing, but you have to remember that MHC2 is seen in APCs, and all other nucleated cells are having class 1 MHC molecule. Clear? What I said, that MHC molecules, just imagine the circle. So now the macrophage, which has phagocytosized a pathogen, chopped it up into small pieces, and it is holding, see, as if it is announcing, see, I am holding a piece of the culprit which has entered our system. Be alert. Up to this, am I clear? Up to this?
Yes, sir.
Sir.
Yes, please. Do you have any question? Hello? Hello? Yes, I think someone is asking question, I thought. No. Okay, fine. So,
Is someone asking question? If you want, you can do it at the end of the lecture, please.
No. I just asked them if they have any problem.
Yeah, I heard. Okay. You can ask also. Someone asking question, feel free to ask. No issues.
Okay. So, a macrophage are dendritic cells or B cells. These are the antigen presenting cells. They have captured a pathogen, chopped it up, and they are presenting it through MHC class 2 molecules. Clear? Now, what will happen? Now, these cells, they are doing two things. Now they are having the pathogen, the piece of that pathogen, and suppose if it's a real sentry, what will do? They will make some sound so that the other sentries all around can be alerted. Here they release some chemicals. We call it interleukins. There are many other chemicals, but here I'm talking about interleukins. So these cells now releasing interleukins. In short, we write them as IL, interleukins. There are many interleukins. For the sake of simplicity, you just write, at present, you just remember one interleukin, very important, that is IL-12. There are many other interleukins like 1, 6, 8. There are many others, but right now I'm trying to focus only one interleukin, that is interleukin 12. 1, then 2, 12. What will happen? See? Oh, I don't have this. Actually, I made a diagram and forgot to save that, and that's why I'm unable to show you the diagram. So it would have, otherwise it had been very easy for you to remember, but unfortunately I made it and forgot to save it. So it's not there. Anyways, if you have any problem to understand, I have to repeat it or I have to draw it through a system. Let me. If you don't understand, just let me know. Okay? So it's not possible right now. So, anyways. So these cells are secreting interleukins. Up to this is the part of innate immunity. So the innate immunity started with I started innate immunity with the skin, and I am ending it up to with the activation of these antigen presenting cells, where it is secreting some interleukins. For now, focus it on the interleukin 12. Up to this is the part of innate immunity. So far, I have not entered into the acquired immunity portion. Now I will enter into the innate acquired immunity. What will happen? The signals are being secreted by the antigen presenting cells. That is here, that is the macrophage. It is having two things. It is displaying on part of that pathogen, and it is secreting IL-12. It will go, and now it is creating a bridge between innate and acquired immunity. There are some cells that is called T cells. What are the cells? T. As you can see here, the T cells. These T cells what will happen is these T cells, those cells who are not specialized. They just like a mold of clay. You have not built any structure out of it, just a mold of clay. So these are called naive T cells. This information will come and will activate the naive T cells. And first they will be TH1, T helper cell 1. Do you get my point? These signals secreted by the antigen presenting cell, consider interleukin 12, it will go and activate a T cell named naive T cells. Then that naive T cell will be activated, and we will call that cell as T helper cells. T helper 1. Okay? Do you understand my point? Hello? Am I clear
Students,
to you?
please respond.
Sorry?
No, I am asking students to respond. Please respond. When sir is asking question, please respond.
My dear students, I'm so much away from you. I cannot see you, and it's not possible for me to understand whether I am clear to you or not. Just tell me, yes, sir, I am clear, or not. Okay, so then I will again repeat it to you. Okay, fine. Yes, sir. Yes.
Sir, can you please repeat this T cells part?
Okay. There are some cells we call T cells. Okay. These T cells, first, they don't know their role. When a baby borns, that baby doesn't know what is its role in its life. Similarly, a T cell, when it borns, it doesn't know what to do. We call that kind of T cells as T naive cells or T0 cells. These are the simplest T cell. They are not specialized. They don't know what to do. What is their role in the immune system? We call those cells as T naive cells or T0 cells. Now, when that signal from the APC cells come and activate these naive cells, they become T helper cell. T helper 1. Okay.
Yes, sir.
Okay. So these T helper
Yes, sir.
cell. Okay, thank you. This T helper cell in turn will secrete interferon, gamma interferon. These are getting little more complicated, right? Just try to remember these things while I'm talking. This T helper cell starts secreting what? Gamma interferon. Gamma interferon. I have already mentioned this T cell part is the part of acquired immunity. That acquired immunity is getting activated by the innate immunity through APC cells. Now it is the time for the T cells or acquired immunity to return the favor. How? As I said, that these cells are secreting gamma interferon. There are many other molecules. I'm mentioning the principal one. Okay. The gamma interferon goes and activate many other macrophages. See, there is a problem. You be careful and try to catch those pathogens which has entered into the body. So far what I have mentioned, a pathogen is entering a cell called antigen presenting cells, capturing it, sends a signal to the acquired immunity, a T cell getting converted into T helper 1, and that in turn is releasing a molecule called gamma interferon, which will in turn activate the innate immunity, again the macrophages. So never forget this structure, that it is like a bridge of between innate and acquired immunity. IL-12 and then gamma interferon, which in turn again activating the other macrophages, or telling about, giving the signal about the infection that has entered, the pathogen that has entered into the system. Clear?
Yes, sir.
Okay.
Yes, sir.
Yes, sir.
Thank you. There is some, now I mentioned about a cell called T0 cell or T naive cells, right? T0 cell or naive T cell. It doesn't know the role in the immune system. So according to the input, it will change itself. First input was IL-12. First input was IL-12. So it got transformed into T helper cell 1. Now, there is some unknown cell, not very clear. There are some definitely some mentions are there. Mostly it's an unknown cell from where IL-4 is released. Interleukin 4 is released. What will happen with that interleukin 4? If interleukin 4 is released, and if this interleukin 4 activates T naive cell, then the T cell will get converted into T helper cell 2. Am I clear? If the T naive cell is activated through IL-12, it will convert into T helper cell 1. If it is activated through interleukin 1 secreted by an unknown cell, then this T naive cell or T0 cell will convert into T helper cell 2. Am I clear to you or not?
Yes.
Please.
Yes, sir.
Okay. This T helper cell 2, what is its role? Ultimately, it will activate B cells. B from bone marrow or bursa of fabrica. Okay? In birds, or in our case, it's bone marrow, B cell. You must have heard the term B cell, T cell and B cell. So, first T cell is getting converted into T helper cell 2, and then this T helper cell 2 in turn will activate B cells. B cells are why B cells are important? Because these are the cells that produce antibodies. Okay? Any doubt?
No, sir.
Okay. So, write it down if possible. I will give you a diagram later on. Right, for this purpose, you write it down. IL-4, then it activates T0 cells, convert it into T helper cell 2. Then from T helper cell 2, it will secrete IL-4, IL-5, and will activate B cells and convert it into active B cells, which will produce antibodies. Clear? These are all part of acquired immunity. Now, here, so far I have mentioned about T0 cells that getting converted into TH1, TH2. There are different other types of cells. Here as a just you can see here TH17, T regulatory cells. They have their own function. T regulatory cell, in a nutshell, it suppresses the activity of T cells so that it doesn't go wrong or the autoimmune disease does not occur. So in the spree of killing other cells, it can harm its body. So that it doesn't happen, there must be some regulation, and T reg or T regulatory cell, T suppressor cells, they are present to suppress the activity or regulate the activity of T cells. Okay, you don't have to remember this, right? You don't have to remember all these things right now. So here you can see, I'm just reading it. CD4 positive T cells. It is written. Two things you have to remember: CD4 positive and CD8 positive. What are these? CD, cluster of differentiation, or a kind of, suppose a protein, protein that are on the outside side of the cells. What type of proteins are there on your surface? According to that, they will be named. CD4 is present in these cells, so they are termed or named CD positive, CD4 positive cells. So which are the CD4 positive cells? T0 cell, T, then it changes into T helper cell 1, T helper cell 2, TH17, T regulatory cells. These are all CD4 positive T cells. Okay? And with whom it is interacting? With MHC class 2 molecules expressed where? On the professional antigen presenting cells. So far what I have said. Now, and another thing I have already mentioned, all other nucleated cells are having MHC1 molecule. At the very beginning, I said there are different types of pathogens. Some are very smart. As soon as it enters the body, it can slither into or inside another cell. So it's not roaming around that macrophage will see it and will destroy it. It is trying to hide itself. As soon as it enters the body, it is entering inside another cell so that it cannot be caught. Right? Am I clear? If not, please let me know.
Sir, it's clear.
Okay. So another pathogen which is coming inside the body, then entering into another cell of the body. So macrophage will not be able to detect it, or APCs will not be able to detect it. But what will happen to that infected cell? Because in that case, what happens, there is another kind of T cells. We call them CD8 positive. With this, I will end. Don't worry, it is getting too much, I know, understand. There is another kind of cell called CD positive, because those proteins are there on their surface. CD8 positive, they are not having CD4. CD4, I have already mentioned, those are T helper 1, 2, 17 or T regulatory cells, and CD8 positive cells are there when they encounter a cell. Which cell? A cell having MHC class 1 with infected cell with MHC1 molecule. Okay? When they encounter such cell and some secretion of IL-2, interleukin 2, you don't have to remember it right now, from that T helper cell, they get converted and become a very ferocious cell. We call them T cytotoxic cell or cytotoxic T cells. What they will do? Simply they will kill the infected cells through different enzymes. Basically, you have to remember this: perforin or granzyme. It is what is like. These cells, cytotoxic T cells, will secrete some highly, just like missiles, it will secrete on that infected cell perforin granzyme, and they will destroy the whole cell and the cell will be destroyed. Okay,
Yes, sir.
is that my point? So today, what's the time now? Yes, it's almost 1 hour. So today I will stop over here. I'm very sorry I have not been able to display you the diagram. Otherwise, it would have been really easier for you to understand. If you have not what I have said so far, please let me know. Okay? With this, I will stop here today.
Thank you, sir. Okay. Dear students, today's talk was really fantastic. You know, this immune system of us is so important for maintaining us in good shape and structure. Okay, and I hope all of you have enjoyed this talk. Now the students are requested to ask questions. I mean, language is not going to be a barrier. If you can ask in Bengali, he can answer in Bengali. If you want to ask in Hindi and English, I am there to help. I am there to help. English is okay, or Hindi and Nepali, I am there to help. Any students, please, you can raise your hand tool in order to let me know that who are asking questions. Is there anyone? Ruhi is there. Okay, one by one, students. First question, Ruhi. Before Ruhi asks her question, I would like to make one announcement. If someone is not comfortable asking question, you can put it on the Google chat box also. That I can also read. And I think sir can also read. And accordingly, he can also answer. Okay, Ruhi, please.
Good evening, sir.
Good evening.
Sir, at the beginning of the lecture, you said that the pH,
Yes.
it somehow it helps in defense. So, as we know, the low pH in the stomach, it helps in destroy of the pathogens that enters through the food. But how the pH, low pH of urinogenital system helps in defense?
See, have you seen ladies catching fish in the pond? Have you seen it? Really?
No, sir. Not.
So,
No, sir.
okay, in photographs also, you will find ladies with nets, or I'm talking about ladies. Why specifically? Because, or you must have seen that ladies are having urinary tract infection more than the male, right?
Yes, sir.
Okay, so imagine a lady with catching fish in a river in waist height water, and a male fisherman is also catching a fish. So what happens? The both of them get exposed to different type of pathogens, but due to the structure of urinogenital system, the females get more, or the opening is much wider. They get more. Uh, they get more exposed to pathogens. How to prevent that? System. The nature has created a low pH, which low pH. What happens in the low pH? The surroundings, it doesn't allow microbes to grow. Do you understand?
Yes,
Just
sir.
the the fungal infections or the many things cannot grow in that low pH, so that the infection cannot, the pathogen cannot survive or cannot grow. That's why the system is there. If what was your question, Rui? Was that your question, or what, or anything
Yes,
else?
sir. Yes, sir. I actually meant that how the low pH helps in defense in the urinogenital system.
Yes, because the low, whether it's inside the body or in the urinary system, whatever, the low pH doesn't allow the microbes to grow properly. It doesn't allow somehow it kills if it is very low, not in the urogenital system I'm talking about, but in the stomach, it kills. Or in urogenital, it doesn't grow, the bacteria or the pathogen doesn't grow properly. It protects from fungus infection as well. Okay?
Yes, sir.
Okay.
Okay. Anyone else? Ruhi, would you please put down your hand so that I can see others? Anyone else? Any student? Especially from third sem? Fifth sem?
I think I'm an excellent teacher. Don't worry. Oh. Yes,
Is Ruhi? Garima, okay. Garima, please go on.
Good evening, sir.
Good evening.
Sir, how immune memory are controlled?
Sorry?
So, how immune memories are controlled?
I have not discussed about that today, okay? So memory cells, that how the B cells memory, T cells memory, it will come after this.
Garima, we are going to have another lecture. And on that we'll be discussing these topics, okay? But if you have any general discussion, general query about basics of immunology, even related to the today's topic, you can ask.
No, it's a very good question, no doubt.
Yeah, it's a good question, but it will be covered in due course of time. That's why I needed to. Yeah.
In the next lecture, I will cover it here.
Yeah, and you will get your answer in next lecture only, because it needs a lot of ground to make before answering this question. It will take a lot of time. That's why. Anything else, Garima?
Okay. So I
possibly
think,
no.
Yeah, there is no more question. Anyways, if you have any question, you can ask me later on as well on the next day. I would request all of you, please read the first chapter of whatever book you have. Okay? The innate and the acquired immunity, the basic, and how innate and acquired system are related. I have mentioned a few of the chemicals, few cells. Please read it once. I think it will help you a lot. If it is you have not studied before, it will help you a lot. I think so. Okay?
Okay, thank you, students. I would also like to request our HOD sir. Yes, HOD sir is there only. So Ritesh da, may I request you to give an official vote of thanks for today's lecture, and then we can move on.
Sure. Okay, thank you, Sanipan. First of all, I must thank you all the students who were present today. Before I go going for the official vote of thanks, the thing which really amazed me today's lecture, the way sir started with the picture of a fort, and he started how this, symbolizing that as a human and how the immune system actually protects that. It was commendable, sir. I was really impressed the way you started with the lecture. And I hope this entire lecture must have been very useful to our students, particularly those who has got the immunology as their paper. And through the course of this vote of thanks, I would like to thank from the Department of Zoology, from the two of us faculties and all the students. Very, very thanks to Dr. Shwamik Agarwal, sir, for being with us and sharing his, drop of knowledge from the vast ocean that he has to our students. And I believe, as I said before, our students must have enriched from this knowledge. My thanks goes to my colleague and the idea developer of this entire monsoon seminar series, Dr. Sandipan Talukdar. My thanks also goes to all my students and especially those students who are helping us in the technical session. There are a bunch of students who has come forward and they are helping with all the technicalities of today's session and sessions going on during this series. And thank you all students. Thank you very much for being with us and having this entire session sportingly. Thank you, sir. Thank you very much.
Thank you, Dr. Vishwa. Thank you, Dr. Talukdar, and my dear students. And I really thank to the organizing committee and the principal of Guru Rathan College for inviting me. Hope I will be helpful to the students in their studies. With this, thank you very much and good night to all of you.
Good night to everyone, sir. Have a very good time ahead.
Then?
Yes, sir. Please.
Okay, thank you, sir.